So, hi, it's Odra, and you are watching Life Sciences from Odra. This lecture is about protein, peptides, and amino acids. This lecture is mostly for M.Sc students, and if you are a graduation student, then it's also helpful for you. So, you can use this lecture for your seminar purpose and for your entrance examination and studies. Today in this lecture we are going to discuss about amino acids and it's the part 1. So proteins are the most abundant macromolecules. They are heteropolymers of amino acids. As you know, there are 20 common different type of amino acids that exist. So then discuss about amino acids. Proteins are heteropolymers of amino acids where each amino acid residue is joined to its neighbor through a special type of covalent bond. The word residue is used with amino acid due to the loss of water molecule during the bonding between two amino acids. There are 20 different amino acids found when protein is hydrolyzed. That means when protein is broken, as the process of breaking or lysis occurs in aquatic medium, so it is named as hydrolysis. So then the first amino acid found is asparagine, and all the amino acids have common names, and these names are named after from where they are isolated, like asparagine is isolated from asparagus, glutamate is isolated from wheat gluten, tyrosine is from cheese. In Greek, cheese is called tyros, so from tyros to tyrosine, okay? And glycine is named so because of its sweet taste. Then discuss about the common structural features of amino acids. All the amino acids are alpha amino acids, as the carboxylic group and the amino group are bound to the same carbon atom, which is alpha carbon. If you don't know, the alpha carbon is the carbon atom which is present next to the prior functional group in an organic molecule. Here in amino acid, the alpha carbon is the carbon which is present next to carboxylic functional group. That's why that is the alpha carbon. Then these amino acids differ from each other in their side chains, which are called as R group. The common amino acids have 3-letter abbreviations and 1-letter symbols. Here is the table of amino acids and their abbreviations and symbols. You can read it. Glycine, abbreviation is GLY and symbol is Z. Alanine, abbreviation ALA, symbol A. Proline, PRO, and symbol is P. Valine, VAL, and symbol is V. Leucine, LEU, symbol is L. Isoleucine, ILE, and symbol is I. Methionine, abbreviation is MET and symbol is M. Phenylalanine, PHEF. Tyrosine, TYR and Y. Tryptophan, abbreviation is TRP and symbol is W. Serine, SERS, 309, THRP. Cysteine, CYSC. Asparagine, ASN, N. Glutamine, GLN, Q. Lysine, abbreviation is LYS and symbol is K. Amino acids histidine, its abbreviation is HIS and symbol is H. Arginine, AGR and symbol is R. Aspartame, ASP and symbol is D. Glutamate, GLU and symbol is E. So you can, when you are writing these names of these amino acids, you can write the full names like histidine, or you can also use the abbreviations HIS. So it's up to you. It's for your simplicity. Then all the amino acids except glycine have a chiral center. If a carbon atom is bonded to four different groups, then it is said to be a chiral center. Here you can see in case of alanine, there are four different groups, but in case of glycine, you can see the carbon atom is bonded to two hydrogen atoms, so this carbon is not a chiral center. Due to the presence of chiral center, there are two different forms of an amino acid which are no superimposable mirror images to each other. They are called enantiomers. So these amino acids are also optically active due to chiral center, that is, they rotate plane polarized light. That's why to specify absolute configuration we adopt D and L system of nomenclature. This system is also used to specify absolute configuration of a simple sugar also. You should remember this. For all chiral compounds, if the configuration of a stereoisomer is related to L-glyceraldehyde, then it's designated as L stereoisomer, and if the configuration is related to D-glyceraldehyde, then it's designated as D stereoisomer. Okay, here in the figure you can see in case of D-glyceraldehyde and D-alanine, hydrogen atom is in left hand side. In both cases, the hydrogen atom has less priority than that of hydroxyl group or ammonium group, and OH group and ammonium group are present towards right side in each case. Okay. Historically, L designation is used for laboratory compounds, that is, the compound which rotate plane polarized light towards left, and D designation for dextro-rotatory compounds, that is, compounds that rotate plane polarized light towards right direction. However, all the L amino acids are not laboratory, and Emil-Fischer convention refers to absolute configuration of all four chiral substituents, that is, where these four substituents are placed in space around the chiral carbon atom. Okay, though amino acids have two forms, but it's awesome to know that all the amino acids residues in the protein molecules are exclusively L stereomers. So in case of bacterial cell wall, there are some D amino acids residues are found very rarely in proteins. Otherwise, all the amino acids are L stereomers. Okay. Then classification of amino acids by R group. This classification is mainly based on the polarity of R group at biological pH, that is pH equal to when 7.0. First one is amino acids with non-polar side chains. Here side chain doesn't participate in participate in loss or gain of protons or participate in hydrogen or ion bond formation. That's why they are non-polar. The side chains have lipid-like properties, so they promote hydrophobic interactions. The nonpolar R groups fill up in the interior of the folded protein as they are present in a polar environment, that is water. Example of amino acid with nonpolar side chains is proline. So proline is a nonpolar R group having amino acid whose side chain forms a rigid 5-member ring structure with alpha amino and nitrogen. Thus, proline has a secondary amino group which is frequently referred to as amino acid. This unique geometry of proline is the reason for the formation of the fibrous structure of collagen. Okay. Now you can see the examples of amino acids with non-polar side chains. They are glycine, alanine, proline, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and you should memorize these things. Then next, amino acids with uncharged polar side chains. They have zero net charge at neutral pH, that means at biological pH. Though side chains of cysteine and tyrosine can lose a proton at an alkaline pH, that means the pH above 7.0 level. But at neutral pH, their side chains are uncharged. Okay. In case of serine, threonine, and tyrosine, each contains a polar hydroxyl group, that means OH group, that can participate in hydrogen bond formation. You can see these things in figure. You can find that there is the presence of hydroxyl group. Then asparagine and glutamine contains carbonyl group, that means C double bond O group, and amide group, that means NH2 group, that can form also hydrogen bond. Okay. The side chain of cysteine contains sulfhydryl group, S, that means SH group. In proteins, two cysteines get oxidized to form a dimer linked through disulfide linkage. You can see this in the figure. The polar hydroxyl group of serine and threonine can serve as a site of attachment for phosphate group. In the figure, here the example of attachment of phosphate group with serine is given. You can see this. So hydroxyl group of serine and threonine and the amide group of asparagine can serve as site of attachment for oligosaccharide chains in glycoproteins. And now the next one is amino acids with acidic side chains. They are proton donors at neutral pH, that is, their side chains are negatively charged. The examples are aspartic acid and glutamic acid. Now the last one, amino acids with basic side chains. The side chains of these types of amino acids accept protons. So at physiological pH they are positively charged, but in case of histidine the side chains may be positively charged or neutral depending upon the ionic environment provided by the polypeptide chains of the protein. This important feature provides the functioning of protein like hemoglobin. Then let's discuss about the modified amino acid residues. Other than the 20 common amino acids, proteins also contain modified amino acids like 4-hydroxyproline, which is a derivative of proline. It is found in plant cell wall protein and in collagen. Collagen is a fibrous protein of connective tissues. And then 5-hydroxylysine, which is a derivative of lysine, it is found in collagen also. And 6 and methyl lysine, it is a constituent of myosin, which is a contractile protein of muscle. Then gamma carboxyglutamate, this is derived from glutamic acid and is found in blood clotting protein prothrombin and other proteins that bind to Ca2 plus ion. Then desmosine, it is a derivative of 4 lysine residues, which is found in fibrous protein elastin. Now, as I'm mentioning the word fibrous protein again and again, you should know that fibrous proteins are made up of elongated polypeptide chains which form filamentous and sheet-like structures. These proteins mainly consist of keratin, collagen, elastin, and fibrin. Keratin found in nail, hair, horn, collagen found in connective tissues like tendon, cartilage, bone, fibrins involved in blood clotting. Elastin is a key component of our extracellular matrix. It also helps skin to return to its original position when it's poked. Then ornithine and citrulline are also uncommon amino acids. They are key intermediates in the biosynthesis of arginine and in urea cycle. The next, amino acids exist as Zwitter ions. Zwitter in German means hybrid ion. Due to the presence of both ammonium group and carboxylic group, they show amphoteric nature. They act as both proton donor, that means as an acid, or an proton acceptor, that means as an base. That's why they are amphoteric in nature. And in the figure, you can see the interconversion between the amino acid and deuterium, and that's it. And the second part of amino acid of this lecture, we are going to upload it as soon as possible. So if you like this, then you should follow the second part of amino acid. Thanks.